Good afternoon, everyone. Welcome to the problem solving session for proteomics course. I'll share my screen now. Is the screen visible to you all?
Hello, yes, ma'am, it is visible.
Okay. So today we will try to address some of the questions depending upon the last week's topic, that is basics of mass spectrometry and sample preparation. So let us see the first question. Narayan has isolated a protein of around 40 kilodalton to test whether the protein isolated is pure or not. He wants to run the sample.
I'm sorry to interrupt. The slide is not changing.
Sorry?
The slide isn't changing.
Oh, okay. I'll stop sharing and start again. Can you see the screen now?
Yes, ma'am.
Yes, ma'am.
Is it changing?
Yes, ma'am.
Okay, so this is the first question. Narayan has isolated a protein of around 40 kilodalton to test whether the protein is isolated, is pure or not. He wants to run the samples on a Mardi-Toff. Which of the following matrices should he choose for Mardi-Toff experiment? So these are the four different kinds of matrices given. So for Mardi-Toff, which matrice should he use? So option A is synapinic acid, option B is picolinic acid, option C is CHCA, and option D is THAP.
Option A,
Option A,
synapinic
ma'am.
acid.
Correct. It's option A. So whenever you are using Malditol, synapinic acid is used as a matrice. So going to the next question. So so 2 to 5 question will be based on the information given here. Just a minute. So Ishan is working on a cell line to identify how a drug X affects the cells. He treated the cells with the drug and palleted down the treated cells. Now, taking this information, we have to answer questions from 2 to 5. So question 2 is he has extracted the proteins from the treated cell and wishes to proceed for mass spectrometry. What is the correct order of the steps he should follow before submitting the protein sample to the central mass spectrometry facility? So option A is tryptic digestion followed by reduction and alkylation followed by desalting of peptides and SDS for quality check. Option B is desalting. then tryptic digestion, then reduction and alkalization, and lastly SDS for quality check. Whereas in option C, SDS is done first, then digestion, then reduction, and then dissolving. And option D is SDH, then reduction and alkylation, then digestion, and then desalting. So, what is the correct order whenever you want to perform a proteomics study using mass spectrometry? So, what are the steps involved? You have a sample of protein. So, what all steps you need to perform, and what is the sequence of the steps that you have to identify from the four options given?
Option A, ma'am.
Option A.
Yes,
Okay.
ma'am.
Any other guesses?
Option C.
Option C. Anyone else wants to try?
Option D.
Okay. We have very varied options, so I think most of you are confused with the sequence of the steps to be done before mass spectrometry. So just remember like this, that you have a protein sample. Okay, so first of all you need to check whether what is the quality of your protein sample. Okay, whether it has different other protein mixture, also whether there are contaminants or etc present or not. So for checking the quality, first of all you need to perform SDS. Okay, so in that manner, first you have to perform SDS. Okay. So now you have an idea about the quality of your sample. Then, so your protein, okay, so it will, you need to perform reduction and alkylation so that the different disulfide bonds and all that will be broken down. Okay. And after that, you will perform digestion. Okay, uh, so you will use tryptic digestion. So you will use trypsin as an enzyme. So that will cleave your protein at different site. And after that, desalting of peptides is performed to remove the salts. So just remember that how the protein will be isolated. Then what first you have to see. First you need to see the quality. Then the protein will be folded in a in its native form. So first you need to remove all those disulfide bonds and all. Only then you can use any protease enzyme which can cleave the protein at different, and you will get the peptides. Okay. So in that manner, you will perform tryptic digestion, and finally you need to remove the salts. So the correct answer is option D. Okay, SDS page for quality check. Then you perform reduction and alkylation. Then you perform tryptic digestion, and then desalting of the peptides. Now going to another question. So this question is also linked to the first question. Suggest the most appropriate control sample he should submit along with the samples prepared in the above question. So here, what is the experiment? He's working on a cell line, and he wants to identify how a drug X affects the cell. So he has treated the cells with drug X and palleted down the treated cells, and that he is using as an experiment. So now, along with that treated cells, you always need a control. Control is a very important in any of the experiment, any biological experiment, because it will it will nullify the effects coming from your experiments. Okay, so option 1 is peptide sample prepared from the untreated cells. Option B is protein sample prepared from untreated cells, maintained simultaneously with the treated cells. Option C is protein sample prepared from untreated parent cells. And option D is peptide samples prepared from cell line treated with another drug Y and maintained simultaneously with the X treated cells.
Option B, ma'am.
Option
B.
B, yes. Option B is correct. Protein sample prepared from the untreated cells maintained simultaneously with the treated cells. So ideally, your control should be designed in a manner that it is gone through the same procedure, same experimental condition as your test sample. Only the thing is that uh, the main test entity. So in this experiment, that he wants to study the effect of drug X. So in case of control, only the treatment with drug X will be missing, but he has to take the same cells, he has to undergo the same kind of experimental procedure. If he's let's say we have to centrifuge for 5 minutes at particular rpm, so then the same thing it should be followed even for the control set. So option B is the correct option. Protein sample prepared from untreated cells maintained simultaneously with the treated cells. Now, coming to question 4, while doing the SDS page, he observed that an intense bend at 40 kD region in the treated samples as compared to the control samples. He suspects that this could be a ribosomal protein of interest to his study. So, which of the following experiment should he do to identify the protein? So these are the 4 options: Western blotting, ingel digestion, 2D Deitch, and in solution digestion. So to identify proteins, what kind of experiment should be used? No guesses? This is very direct.
What about Western blot?
Yes, Western blotting, we can do that whenever we want to identify a protein. See, dyes we have
Solution.
already studied earlier, right? It will give you an information about differential expression in your 2 different kinds of sample, but it will not give you an identity of the protein. You cannot identify a protein based upon uh, 2D dyes, right? Now another information is you are all is already doing SDS page. Okay. So mass spectrometry will also give you an idea about what your protein is. So depending upon the sequence of the peptides, what is the sequence, from that you can identify your protein, right? So you can use mass spectrometry. And here you're using SDS phase. So it will be easier for you to use ingel digestion followed by mass spectrometry. So in solution digestion followed by mass spectrometry, this option is also ruled out. So the correct option is Western blotting, and another one is ingel digestion followed by mass spec. So going to next question. So this question is related to mascot. which is a software used for proteomics sample analysis. So I hope you have some idea regarding mascot. Can I get some response?
Some kind of
Okay. So here, suppose Ishan did a mass spectrometry and now he has a mass list. Okay, so he has now MYZ values in a text format for the peptides of this protein of interest. Okay, so now how will he analyze this data using mascot server? So we'll just go to the link and see what are the options available. Can you see the screen for mascot?
yes, ma'am.
Is it visible?
Yes.
Yes, ma'am.
Okay, very good. So see, using this mascot software, you can use different kinds of analysis. So one you can see here, peptide mass fingerprint, and another one is sequence query. Then it is MSMS ion search. So what, depending upon what is your purpose or what is your query, you need to select any of this function using mascot. So in our question, what is asked, Ishan did a mass spectrometry and now he has a mass list in a text format for the peptide of this proteins. Okay. So we will see what information is given here. So, in case of peptide mass fingerprint, the experimental data are a list of peptide mass values for the digestion of a protein by a specific enzyme such as trypsin. So, this is what we want to see here, right? He has a value, mass list value for the peptides. So, we have to perform PMF. So you can see here the information is given that what are the functions of this different kinds of search you need to perform. So in this question, it is about peptide mass fingerprint or PMF. Just a minute. So going to the next question, what are the advantages of using chip based nano LC? So these are the different options given. Very low sample volumes needed, very tolerant to salt contaminants, no need for sample preparation, and very high reproducibility in the results. So what is the advantage of using chip-based nano LC?
We need very low sample volumes, and also there are no problems of leakages.
Correct. Yeah, you're right. We need very low sample volumes whenever we are doing nano LC, chip-based nano LC. So option A is the correct option. We need very low sample volumes and uh, yes, so that is the correct option. Now going to another question. So here is the diagram given for triple quadrupole mass spectrometer. So you just have to identify what is the role of Q2. So option A is fragmentation of selected product ions. Then option B is it acts as a mass filter for parent ion. C, it acts as a mass filter for product ions, and option D, the fragmentation of selected parent ion occurs here. So what is the correct option? What is the role of Q2? What is the role of this Q2 here?
Is it option D, ma'am?
Option C.
Option, sorry,
C,
Option
ma'am.
D.
A. Option A. Okay. Any other guesses?
Option
Option
D, ma'am.
C.
Option B or option C?
C.
Option C. Okay. It acts as a mass filter for product ion. Any other guesses?
Option D, ma'am.
Option D. The fragmentation of selected parent ion occurs here. Okay. Now anyone wants to, anyone's think it is option B. It acts as mass filter for parent ion. Okay, so we'll see. So you can see from the diagram. So this is triple quadrupole. Okay, so from the LC, the sample will go here. So it is electron spray ionization. It's a ionization technique. Okay, so after ionization, it will go ahead, and here in the Q1, you can see here it's the samples are dense and then they are getting reduced here. So Q1 will act as a mass filter for precursor ion or the parent ion. So parent ion or precursor ion, it's the same. So only the targeted precursor ion of your interest will be selected. So you have to give the machine your uh, target protein or target MYZ value, and only those precursor ion will be selected. Then it will pass to Q2. Okay, so what will happen in Q2? So here it's a collision cell. So as the name suggests, the precursor ion, it will be fragmented here into the product ion. Okay, and then in the Q3, the product ion will be selected, and then the detector will detect, and then you will get the signal in. Okay, so the correct option is the role of Q2 is the fragmentation of selected parent ion. Okay. So any doubts here? So I hope this is clear now. So this is very basic mass spectrometry and how this how the precursor and how the signal is generated. So that is depicted here. So you should have an idea that what is the role of Q1, what is the role of Q2, and what is the role of Q3, and what does detector do, and where the ionization occur, what is the role of LC and all. Okay. So I hope this mass spectrometry, this diagram is clear now to everyone. Should I move to the another question?
Yes.
Ma'am.
Okay, fine. So this question is also related to mascot software. Okay. So as mentioned earlier, mascot is a software used for proteomic data analysis. So here the instruction is given, run an MSMS ion search on mascot server using the file given in the following drive link. So I already have downloaded the file. So if you all want to try, what we can do is that I can share the link in the chat box uh, of the software as well as the link to the file, and then taking this information, you all can try doing this in your computer. So is it fine for you all? So that will also help in You can also do it. It will be a practice for you also.
Yes, ma'am. Sure.
Yes, ma'am.
Sure. So I'll share the link. Right, So I have shared the link with you. So one is the link for the file. So first go to that link and download the file and then open the mascot software and then let me know. Then then we'll see the question.
Can you please show the question?
Sorry?
Can you please present the question that was asked?
Yeah, sure. Just a minute. Yeah. Can you see? Can you see the screen?
Yes, ma'am.
Okay. So everyone has downloaded the file. Anyone left?
Ma'am,
Yes.
well, there has been a problem in downloading the file.
Okay. You can just click on download and it will download. I mean,
Ma'am, it was downloaded but could not support the format.
yeah. So that is a different format. The software and mascot will support it. Okay. So you
Okay,
just press the download and just save it on a location wherever you want to save it on your computer. And then while doing this search on mascot, you just give that location. Okay.
Okay, ma'am. Thank you.
So here all the parameters for MSMS ion search is given. So first of all, if you all have downloaded the file, then we need to go to the mascot software. My entire screen. So this is the mascot software, it's the server. So we need to perform MSMS ion search. So this will be based upon identification based on raw MSMS data from one or more peptides. So the file that you all have been given is the raw MSMS data file of any X peptide. Okay, so we need to see from the databases that what is the identity of this peptide, what kind of protein class it belongs to, and all those information we will get from the databases. Correct. So we will start our MSMS ion search. So first we'll see so these are the different parameters given. So first of all, you all click on perform search. All of you are doing this. Just a moment. So all of you are on this page.
Yes, ma'am.
Okay, fine, correct. So here you have to enter your name, your email ID, and if you have not signed in, it will ask for that. So just mention your email ID which you are using. Then we need to give some title. So databases is basically you have to select from here different databases. Okay. So this contaminants you have to default select it because there could be some contaminants already I mean present, and so this as a default you should always select, and this is depending upon different class of organisms, the different databases are created, and so we will select Swiss prot. So we'll see what is given in our so here the database is Swiss prot. So you all select here Swiss prot. Then taxonomy is all. Enzyme use trypsin. Quantitation none. So you all can you see the details? I mean the small parameters that are mentioned here.
Yes,
Yes,
ma'am.
ma'am.
Okay. So fill these parameters. If you come across some query, let me know if you are not able to do it. So modification here you have to give this one. Here you have to give oxidation, peptide charge. So here you have to give your file of interest. So everyone has selected different parameters as mentioned here.
So, ma'am, I want to ask what is
Yes.
decoy? What is decoy, ma'am?
Decoy. Okay. Uh, so I don't remember this parameter currently. I'll see to it and let you know in the next class. Okay.
Thank you.
It has something to do with uh, data analysis, how the match is being done in. So here you are matching it with the databases. Reference libraries are available for your MSMS sequence data. So it is a kind of match. But exactly what is the procedure for decoy, that I'll let you know in the next class.
Ma'am,
Yes.
ma'am, in this amino acid section, what should we select?
Where?
Ma'am, in this amino acid section, after the database.
After the database. Here. This one.
Yes, ma'am. Yes, ma'am.
So this is the list of the database. Okay, so from here you have to select the database and then it will come here. So I'll just show you. Okay. So if you can see this, I have. So can you see this is mascot is open now, uh, or is it the PPT only you are able to see?
We can see the mascot page here.
Okay, so here contaminants is already selected. Now you want to select Swiss prot. Okay, so you just have to select here Swiss prot and then press this and it will come here. So this is the list of all the databases available with mascot. In this manner you have to uh, so same here, different kind of modifications are given and you have to select that for fixed and variable modifications. So have you all selected all, entered all these parameters? Can I get some response?
We can.
Okay, so if you have entered all these parameters, then you can just do start search, and then it will take a few minutes, and then you will get the result.
I have got the results.
Acha, nice, very nice. Everyone else, have you got the result? Okay, so we'll move to the questions now. So these are the questions given depending upon the MSMS search that we had performed using mascot. So what is the approximate score for the top hit? So if your search is done, you will get a list of proteins by which your sequence are matching. So top hit list will be given and the score will also be displayed. So if you have done, you can select from these four. And then the name of the protein will also be given. So what is the name of the protein with the top hit? So the first hit that you are giving, so it will give you some 10-20 list of the proteins to which your sequence matches. Okay, so what is the top hit that you have to select here? And what is the species or organism this protein belongs to? So all this information you will get in your result.
And in the like the protein name is chlorophyll AB binding protein 1. That
Yes.
is,
Okay, correct.
and the organism is Synapis alba.
Correct.
And but the profit is not coming the same. In my laptop, it is showing 276.
Okay, so it is near to this, which is fine. So we'll see. So if you, so all of you are getting this, so it is somewhere near 200 only. You are correct. So all of you got this result, or you want me to perform this MSMS, and then I can show you also.
Yeah, could you show it to us?
This one I told you about.
Yeah. So decoy, what other parameters? Okay. Submit the file. This is the file ATP21 MSMS. We'll start the search. So I have to just verify my email ID. So just give me a minute. I'll share the screen again. Unfortunately, I had entered the wrong email address. There was some error. That's why the error was coming. Start the search. So can you all see the result?
I guess you didn't select a decoy over there.
Okay. I did select. Okay, here the Swiss prod was not selected. Some of the parameters were changed, I guess. Right. Yeah, so you can see now. So this is the matches protein families. So this will show from the databases what all different proteins your peptide fragment matches to. So the first one is chlorophyll AB binding protein. So as someone has mentioned, this is the top hit and this is the score. 276 is the highest score. So that is not mentioned. The range is incorrect, but it is near to 200. The second question is name of the protein. So here you can see the name of the protein as well as the class is given, Synaphyllus alba. So that is the class and the name is chlorophyll AB protein, AB binding protein. So in this manner, you can just from the peptide sequence that you get from your mass spec data, you can find out what is the potential pro, kind of protein of your particular, I mean, from the sample. So, because we have the information, there are lots of databases of different organisms and species, and from that you will get an idea that what kind of protein is yours. So, whenever you are doing a proteomics experiment, let's say you are doing some biomarker discovery or you are seeing a drug effect of drug, and then you see some different kind of protein upregulation or downregulation. So at that time, you have no idea about what your protein is, right? So you need to identify also your protein. So for that, this tool is used. And from your peptide sequence information, so we have different databases, and that will give you information that what is the match, and the scores tells you how closer your protein is to this particular the match that is given, the hits. So higher the score, better is your match. So this is like 33. So this is least matching to your protein of interest, to your protein that you have performed mass spec, and this is here the score is 276. So this is the first top hit, and so your protein is showing more much closer sequence similarity to this protein. Okay, so this is the last question for today's session. Thank you all. And
Ma'am,
yes,
so ma'am, there is a question in our assignment.
huh?
So basically, they have asked, they have told that there is a peptide sequence from our profit, and they have asked ki how many fragments ions were used for scoring. So by using this data, can we like interpret what they are actually asking? They basically they told us that there is a topic like
So,
this
so they are asking what is the top, what is the question?
question they have mentioned they from the top bits
Mhm.
they have mentioned this peptide sequence. Okay, and
Okay,
they have asked us that how many fragment ions were used for such scoring.
okay. So for that, so I need to see the question. Only then I can, I mean, comment upon that. So basically, the software will be using different. So something like this will be there, I guess. So
And this is
software will be using some matches. So here it is showing not assigned. So these are the matches that are not assigned. And from here you have to see. So this is for not assigned. In your case, it is for the assigned ones. So here it will give you an information for what are the, so you can see these are the fragments that they were looking. This is label all possible matches. Label matches used for scoring. So this is red. This one is used for scoring. Y, the red one. But here what I have showed is for not assigned. And I guess in your question it is asked for uh, what is assigned, right? It is vice versa. So in a similar way, you have to see for that.
is there any options of like assigned or something?
Uh, options for what?
You asked now for assigned or unassigned.
Mm-hmm. Yeah, so I selected it from here. So these are unassigned. So similarly, like here it is non-significant and here it is unassigned. Okay, so this is completely changing. So maybe that question, it's in the assignment. So once it is, once the deadline is done, only then like we can discuss that after that. So since the deadline is not over, you have to try to figure out that by yourself. If you have some specific doubt, like a certain part of the question you are not at, like you are not able to understand, then maybe you can send that on in the Google form or one form is floated. So in that we can address that. Any other questions? What is the deadline for your week 6 assignment?
It is 7th September.
Okay. So what you can do is you can mention a particular section or whatever you are not understanding in the question, and then we'll try to revert back to you and try to solve your query. Okay. So since it is already in the assignment, we won't be discussing it now. Once the deadline is over, then we can discuss.
Sure.
So any other queries, or should I end the session? So I'll stop sharing now.
Actually, ma'am,
Yes.
there's a query regarding the exam. So can I just ask you like?
Okay. So regarding exams and all, so I that will be the timing and all will be given to you. What is the dates and all that will
Actually,
be.
not the timing, ma'am. Basically, in the exam registration format, they have asked about the credit transfer. So I have made it yes, but I got my final result of my post graduation. So is it really valid for the credit transfer to me?
So that for that you need to write directly to the NPTEL team. So because there are specific people assigned for that particular thing. So you can write an email to NPTEL that I have this query regarding the examination and this. So they will address that because I do not look after this. Why do I would not be able to comment upon that.
Thank you.
Everyone, we will meet next Saturday for our week 7 session. Bye bye.
Thank you. Bye.